Hello everyone. So let us get started the next chapter, that is on body fluids and circulation. A very important chapter. You have got lots of concepts that has to be covered. So for your NEET exam, so if you take the entire human physiology unit, you can expect 20 to 25 questions. And on an average, a single chapter can get you 12 marks. That is, you can expect 3 questions from each chapter. From body fluids and circulation, the important high yield topics, if you ask me, the first will be your cardiac cycle, and they can ask you a question definitely on ECG. So these two appear every year, and ECG is almost a definite question. You cannot escape from ECG, right? And the third question on blood grouping. So all these topics have been put as a separate video. I'll be discussing in the upcoming videos. And apart from this, they can ask you a question on plasma proteins, or they can ask you about functions of different cells. They can ask you especially the functions of WBCs. So the topic 4 and 5 is what we are going to see in this video. That is the general introduction on blood and its components. So try to cover this chapter under these headings and try to solve more questions on these topics. So the purpose of me starting off by giving you important topics is just to utilize this for your last minute revision. So during your last phase of revision, it's not possible to know just sit and go through every single concept or every single question. So it's better if you just focus on questions from these topics so that you can score maximum marks, right? So that is the main objective of giving you the important topics, and it is not to just convey that just focus on these topics. You need to study every single aspect of NCRT, but for revision is you can probably skip few topics and probably you can look at high yield topics. So that is the main objective of giving you a list of topics. So let's get started. The introduction, that is on blood and its components. So talking about it, so you all know blood is a connective tissue, right? So it is a connective tissue, and if I take an 100 ml of blood in a tube and I try to centrifuge it, I will be getting two components, two main components. One will be deposited, and that is called as formed elements, and the another layer is plasma. So my blood, broadly, it contains two components. One is plasma, another one is formed elements. Formed elements, in turn, if you ask me the list of formed elements, it is RBCs, WBCs, and platelets. All these are some basic information you all know. So you do not want to bore you with explaining this again and again. So it contains three components. And plasma, if you take, it contains lots of electrolytes. So it contains water, it contains electrolytes, it contains proteins, and other substances like your clotting factors. Almost every single substance that flows in the blood is going to be related to the plasma. So almost all substances that travel is going to be a part of plasma. So for every 100 ml of blood, 45 ml is going to be formed elements, and remaining 55 ml, or I can say it as percentage because I have told for every 100 ml, right? So instead of having it as ml, let me make it as percentage. So you will have 45 percentage of your blood is composed of formed elements and 55% is composed of plasma. Now, we are just going to discuss few points about each of these formed elements and also each of the components of plasma. So let us start with plasma because it is like very simple. We don't have much points to discuss there. So when it comes to plasma, you should know that 90 to 92% is water. So predominantly if you take it is made up of water, remaining about 6 to 7% is made up of proteins. So other substances are all no negligible in numbers. So we are not going to take that into consideration. So two main components, if you ask me, I will just tell is water and proteins. Now, talking about proteins, that was a NEET 2018 question, and it is also a frequently appearing question in your model exams, and it has appeared even in AAPMTs before. So, you just have to know that plasma contains many proteins, but three important basic plasma proteins are albumin, globulins, and then fibrinogen. Like these are the pillars of plasma. If you take albumin, what is the function of albumin? Now, you all should understand a terminology called as osmotic balance. What is this osmotic balance? See, you take food, it is getting digested and it is getting absorbed. So all your food products that are no getting digested and absorbed are entering into bloodstream. And from the bloodstream, so suppose let me take this is a blood vessel and this is my tissue. So from the blood vessel or from the bloodstream, the substances are going to enter into the tissue. And I have told you, any substance that is going to move inside our body is going to be guarded, or it is going to operate under a gradient. Right? So when I say movement of substances, it is going to be by means of a concentration gradient. And the process is called as osmosis because every single cell is made up of some cell membrane, and that is going to be semi-permeable, right? So it is going to be an osmotic process. So every time substance cannot move towards a particular site. For example, not all substances every time can continuously get into the tissue. So tissues will be completely then no being in a toxic state, right? So there has to be a cutoff, or there has to be a regulation for the movement of these substances. So only when the gradient is created, or when there are other pertaining factors that can promote osmosis, the movement of substances can take place, right? So in order to maintain that gradient, in order to regulate the flow of substances from the bloodstream into the tissues, there has to be a balance between the concentration of the substances both in the bloodstream and the tissue. So such a balance in the concentration of substance, which in turn is going to put a check on the osmotic process, is called as osmotic balance. So this albumin is going to accordingly manage the osmotic balance in the body, that is between the blood vessel and the tissue. So when somebody asks you what is osmotic balance, you need to have this much of understanding. Okay, there is a movement of substance. So in order to control the movement of substance or put the check, there has to be a osmotic balance. It has to be accomplished. Osmotic balance in the sense in terms of concentration. So what is going to help in that? It is going to be albumin. Globulins, I hope everybody know we have got a major type of globulin called immunoglobulins, right? So globulin in general, 99% it is going to help in the defense mechanism. So it is going to help in the synthesis of antibodies, that is immunoglobulins, and that in turn is going to help in the destruction of the pathogens and other foreign substances. Fibrinogen helps in clotting of blood. So what happens? How the blood is going to clot? What is the mechanism of coagulation of blood? So that can also be asked in a NEET exam. It is a frequently asked 2-mark question in your school exam. So if you have body fluids and circulation in your portions, then you can very well expect this question. what is the role of fibrinogen? So I told it is going to help in coagulation. So how it is going to help in coagulation? That is very important. So what will happen is whenever there is an injury and there is bleeding, there is a protein called prothrombin. Pro means I have told pro or any substance that ends with GEN. This is zymogen or inactive form, right? So what is going to happen is whenever there is an injury, there is bleeding, this prothrombin from the liver is going to be released into the bloodstream and it is going to be activated to something called thrombin. So this step is going to be aided by an enzyme called thrombokinase. So what happens? First, there is injury. Following injury, there is activation of thrombokinase that in turn activates prothrombin to thrombin. Now, fibrinogen. So it is a zymogen, right? It's an inactive protein. So this thrombin is going to convert fibrinogen to fibrin threads. And this fibrin is what is going to form the clot. So say suppose this is my site of injury and you have blood leak from this site. What will happen is you will have lots of platelets that are going to come around like this. Okay, so there are a group of platelets that no just comes around. And all these platelets has to be tied like a big knot. Okay, just imagine there are some 10 thieves coming into your, they are breaking into your house. So you are Bruce Lee or whatever. So you just try to tie them on a pillar or a wall. So what do? You bring all those thieves together and you're just going to have one single thread that is going to be knotted. So the same thing here. So this fibrin is going to allow all the platelets to get aggregated and form as one single big clot. So that clot which I'm talking is actually nothing but a fibrin threads that runs like this. So something is just going to knot all the platelets that are come to the site of injury. So this is clotting. Okay, so this is how fibrinogen is going to help in clotting. So it is not directly going to act, but in turn it has to be activated by a substance called thrombin. That in turn is getting activated from prothrombin by action of thrombokinase. And this fibrin is going to be like a thread-like structure that is going to aggregate all these platelets. So this is the function of plasma proteins. Now coming on to formed elements, I have nothing much to discuss. RBCs, you all know the basic points. That is graveyard of RBCs. You have to know it is spleen. So what you need to know is all your formed elements, be it RBCs, WBCs, or platelets, everything is going to originate from bone marrow. Is going to originate from bone marrow. And the process of formation of all these formed elements, I generally call it as hematopoiesis. So hemat means blood related cells. Poiesis formation. Okay, hematopoiesis. So if the formation of RBCs I called as erythropoiesis. WBCs I called as leukopoiesis. So just by the name. Okay, so I hope you all know the medical terms for RBCs, WBCs, and platelets. Now, what we need to discuss is about classification of WBCs and the functions of each of those. This is very important. So, talking about leukocytes, we have got broadly they are classified into granulocytes and agranulocytes. They are broadly classified into granulocytes and agranulocytes. Now, what is the meaning of a granule? So say suppose this is my cell. Inside my cell, I have got cytoplasm. If the cytoplasm has got lots of laddus like this, then I call it as granules. So basically granules are going to contain the important substances that is going to be released by a cell, right? So those are called as granules. So granulocytes, if you take, they are going to contain granules. Agranulocytes, they do not have any granules. This is the basic understanding. So depending on the morphology of the cytoplasm of the cell, it is being classified into granulocytes and then agranulocytes. So granulocytes, if you take, we have got three types of cells. that might come under granulocytes. So they are acidophils, or I call it as eosinophils. Functions of all these are important. Next, basophils and neutrophils. Agranulocytes, if you take, monocytes and lymphocytes. So if you observe, all the cells that has a suffix called "phils" are granulocytes, right? So they have got a suffix of "phils," and all your sites are going to be agranulocytes. So acidophil, basophil, neutrophil, monocytes, and lymphocytes. So functions of these are important. Um, the first important point that you must know with respect to formed elements is neutrophils are the most abundant form. So if suppose I take patient's blood sample, I try to view it under a microscope. If the patient is normal, like he is not anemic or do not have any blood disorders, blood cell disorders, then in the smear, in the microscope, majority of the WBCs that I observe will be neutrophils, and least form will be basophils, right? In a normal patient, mostly even I will not be able to identify a basophil, or maximum I'll be able to identify only one basophil. So almost 60 to 65%, or sometimes even 70%, you will have only neutrophils, and it is 0 to 1%. Others will be found intermediate, like acidophils. If you take 6 to 8%, lymphocytes 20 to 25%. So the second most abundant will be lymphocytes, because they are part of immune component, they can be found, and 6 to 8% you will have acidophils. Not much to take home point. Just know abundant is neutrophil, least is basophil. Functions of these are important, like acidophils and basophils. Just remember, like A for A. Acidophils, they are involved in allergic responses. So I hope you all know what is allergy, right? So no, it is basically an exaggerated immune response to some normal components like pollen grain or dust. All these, if you take, no, there are some people who are allergic to dust, meaning they start sneezing, coughing, and they show some reactions to certain particular allergens. So those are called as allergic reactions. So acidophils are the main mediators of allergic reactions. Basophils, if you take, they are mainly responsible for inflammation. So whenever there is an injury, there will be an inflammation that will happen. So for that inflammation, basophils are going to just come into the site of no site of either a location of a pathogen, or there can be an injury. Wherever there is a foreign substance being landed, there you will also find basophils and acidophils is going to help in the inflammatory reaction. So how the inflammation is going to be mediated is, I told you all these are having granules, and all those granules have some substances specific for each of them, right? So this basophil is going to secrete some inflammatory substances like serotonin, histamine, etc. That is going to aggravate the inflammatory reaction. So that's why, if you take some if you see some inflammatory reaction, we give this antihistamines, like these lotions. I forgot the name of the lotion, common name. So we give this lotion whenever there is this itching or redness which is happening for no cause. Then we try to go for some lotion, right? Inflammatory, anti-inflammatory lotion. All these will have antihistamine, anti-serotonin preparation, because all these are the cause for for inflammation, right? So I'm just trying to inhibit the causative substances, and thereby I'm trying to reduce the inflammation. Now coming on to monocytes and lymphocytes. Lymphocytes, you all know, it is going to involve in our immunity. So this, I do not want to confine your knowledge of lymphocytes by just saying there are two types of lymphocytes. As of now, just know immunity, and then it is going to help in the production of or developing immune system. That is fine. But you have got lots of details to know about it, that we will be dealing it in 12th grade in human health and diseases. Okay. And monocytes, if you take both of these neutrophils and monocytes, they have got a phagocytic action. And These monocytes, we actually call it as tissue macrophages. Now, the understanding behind this is, why am I telling neutrophil and monocytes both are having phagocytic role? Neutrophils, they are the first phagocytic cell to know actually um, get into the site of injury and try to kill all the pathogens. Now, the concept here is, following entry of any pathogen or a foreign substance, we have inflammatory reaction, I've already told. And during this inflammatory reaction, it is a protective response. All these neutrophils will try to eat up all these foreign substances. If the causative agent for that particular injury or infection is going to be there for a long time and it is trying to enter into tissues also, then we have got monocytes that gets its role. So initial phase of injury, you have got neutrophils that are going to be phagocytic. Later stage, mostly in the tissues and all, we have got monocytes. So tissue phagocyte or tissue macrophage, I can very well call it as a monocyte. And first level phagocytic cell is going to be neutrophil. So first neutrophil followed by monocytes. Just remember like this. These are the functions of different types of WBCs. And platelets, common name, thrombocytes, um, and then it has got some 1.5 to 4 lakh cells can be found in per cubic millimeter. It is going to help in blood clotting. We just saw in coagulation system. Nothing much significant to study under platelets. So from this video, you just have to take down the functions of different plasma proteins, and then you just have to understand the different functions of WBCs. Sometimes, very rarely, if the examiner wants to give you a simple question, he asks you, what is serum? Serum is nothing but plasma minus clotting factors, right? So I told you plasma is the place where you will find almost every component, electrolytes, clotting factors, and all that. So when I thought about coagulation system, we just saw a few steps. And apart from all that formation of fibrinogen to fibrin, it is not just the thrombin that will come and help. You have got lots of other factors called coagulation factors or your clotting factors. You have got about 12 factors present in the plasma that will help in the clotting. So if you have the blood outside for some time, it will clot because of presence of clotting factors. So you just have to remove the clotting factors for the blood to stay uncoagulated for a long time. When you will leave the blood uncoagulated for the long time is during your laboratory. This is a application of serum. Now, if suppose you try to get a blood report of your father or mother or grandparents later, they would have done some tests like random blood glucose levels. There they would have taken plasma glucose value. But if any of your family member is a thyroid patient, serum TSH, serum T3, T4, or if any of your family member has cholesterol, serum LDL level, serum HDL level. So for some quantities or for some components, they take just the plasma, which means what? They draw the blood from the patient and they're just going to preserve as it is. But for some cases, for some components, they try to remove those clotting factors because they need the blood for the longer time. They're going to have it for a longer time, so they're just going to remove the clotting factors. So out of the blood or from the plasma, if I remove the clotting factors, it is called as serum. If I maintain as it is called plasma. Right? This is sometimes becoming examiner's favorite. Right? So just know this point. With this, I would like to put an end to this video. I'll meet you in the next video wherein we'll be discussing about the blood grouping. Thank you all. Tata. Bye.